Hi, everyone. My name's Dan Lock. I'm a clinical scientist based in Leeds in the UK at a large hemato-oncology centre there. Thank you very much for the invitation to speak here today. So I'm just going to run you through a validation we've completed recently for a high-throughput NGS screen to investigate the key driver mutations that we find in the myeloproliferative neoplasms. Just leave that disclaimer for a second. So a quick overview for the talk. I'll give a little bit of background on the molecular pathology of the myeloproliferative neoplasms, as I'm aware not everyone will be necessarily familiar with those. Talk about our rationale for the NGS panel that we validated, and then a little bit about how we went about that validation. I'll give an overview of the service design, because obviously we're a clinical centre, so this has to meet the needs of patients. And then I've got some 6-month data. We've been live now for 6 months, so I've got some audit data looking at how the assay is performing. And then I've got a few interesting case studies that I thought I might go over with at the end. So the myeloproliferative neoplasms, the clue's in the name, it is an excess of cells in the periphery. So either an excess of red blood cells or an excess of platelets, and less frequently an excess of connective tissues in the bone marrow. These cells are terminally differentiated, so it's slightly more manageable in the clinical setting than some of the more acute diseases you might see in the hemato-oncology setting. And it's characterized, like I say, by an excess of cells. So here we can see the excess of platelets in the peripheral blood in essential thrombocythemia. And on the right, this is a marrow, which is packed, and you can see some clusters of large cells there, which are the megakaryocytes that produce the excess of platelets. The molecular pathogenesis is underpinned by mutations in 3 key oncogenic genes. So we've got the JAK2 gene, which we see mutated in about 95% of polycythemia cases have a JAK2 mutation. And then in essential thrombocythemia and primary myelofibrosis, we see mutations in CALR in exon 9, where we see 52 base pair deletions and 5 base pair insertions. And then in MPL exon 10, these are fairly rare, but these are activating mutations at 2 principal hotspots. We see multiple substitutions here, which is why we needed a sequencing solution for this. But it's nice because it's a fairly small panel and we've only got 3 key genes that we're investigating. So a little bit of background, the driving forces behind consolidating our testing onto a small NGS panel. In 2020, Genomics England launched the new NHS Genomics Medicine Service, and one of their advisory points on that was that we consolidate all of the testing in specialist hemato-oncology centres, where not only you could achieve some sort of cost benefit by bringing all of your tests onto a single site, but also you could standardize the service for your patients, and you could bring all of your experts into one centre. So I work in Leeds, and we've now taken on the work from Newcastle and Sheffield. We service about 18 million patients across the northeast of England. So there's a big driving force at the moment in the NHS to move on to small panels, which means we can retire some of the historical tests that we've got. The way we've done it previously, the standard of care testing, because that JAK2 mutation was so prominent in the disease types that we've got, we would have an allele-specific PCR-based test for the mutation. So you would get amplification if the mutation was there, and you wouldn't if not. And similarly for the CALR 5 base pair insertions and the 52 base pair deletion, we had allele-specific tests, but these are very targeted. The less frequent mutations that we saw in MPL and exon 12 required a sequencing solution, and at the time all we had available was Sanger sequencing. So we would have a sequential testing strategy, and if it was negative in the peripheral blood for those patients for the JAK2 and the CALR, we would then request a bone marrow biopsy from those patients because of the reduced sensitivity for the Sanger sequencing to test for the less frequent exon 12 and exon 10 MPL mutations. That wasn't without its faults, though. So we know that from experience, we picked up in 2021 a false negative result for a patient who, when they later went on to have full sequencing on our pan-heme oncology panel, was discovered to have another mutation slightly downstream at position 615, which we suspect was actually disrupting the binding of the primer, which returned a false negative in this example. Additionally, because we had multiple sites performing multiple tests, we had disparate turnaround times for the patients, and because some of those tests sequentially would require different tests, like Sanger sequencing for MIPL, for example, that meant we had different turnaround times and different limits of detection. So the patients potentially with the same disease, but a different mutation, would have a different time to diagnosis, and some would require bone marrow biopsies to enhance the sensitivity of the testing strategy. So it wasn't without its flaws. In order to consolidate the service, we considered that we would stick with our standard of care test, but that's got the problems I've already described. We did also consider consolidating to a single site with the sequential testing still in place, but that still has the problem of the different turnaround times. So it was decided after an options appraisal that a small NGS panel would be the right way forward for the patients. We knew that across the region, within that 18 million patients, we would expect to receive approximately 130 samples per week. So we needed a high throughput approach. And the NGS panel would allow us to standardize the turnaround time and the limit of detection and bring it onto a single test. We did have some concerns moving to a sequencing solution that there would be, we would have to demonstrate that the additional cost was worth it for the patients in context. And also because this is amplicon-based or PCR-based sequencing that we're doing, there is always background noise in your sequence data. And the current recommendations for a limit of detection for that JAK2 V617F mutation is 1%. So it was very important to us that we didn't see any background noise above 0.5%, because otherwise we had the potential to end up with false positives. And when we performed the options appraisal, we weighted it so that the performance of the assay was the key component that we were interested in that we ended up with the best assay for our needs. So a little bit about the design of the panel. We've got the three hotspot targets that I've mentioned there: CALR, MPL, and JAK2. There is a very rare entity, the CSF3R variant 618, at position 618, sorry. And we only see about 1 or 2 of those per year, but those patients do have a very long time to diagnosis. So we thought we would include that too, because it is a diagnostic variant at that position. So we've got those 4 key targets in. We have also included, you'll see a bunch of other amplicons there. They are for a different disease type, chronic lymphocytic leukemia. Now, the assay that we validated, we can actually run 200 samples per week. But we were only expecting to get 130. So to improve the efficiency, we've included these additional targets with the intention that eventually we can put some of our CLL work onto this panel as well. But principally, we're looking at these 4 key targets that I've got listed here in red. As many of you know, before you conduct a validation of a new NGS panel, it's worth sitting down with your colleagues and working out exactly what your acceptance criteria will be. So obviously, the limit of detection was very important to us. We included samples to ensure that we had variants within each amplicon of TP53, for example. The indels that we see in CALR, the largest deletion, well, the most common is 52 base pairs, but we have seen deletions up to 83 base pairs. So we really wanted to put it through its paces and make sure that we could pick up some of the larger indels that you see in CALR. We were very interested in the wild type background, because obviously that's going to affect your limit of detection. And then we put it through its paces by including some suboptimal quality samples. We're receiving DNA from various sources, and some of those can be particularly low yield, especially where the bone marrow is fibrotic and you don't get a lot of DNA out, you don't get many cells out from the sample. And we also put some validated control samples through to investigate the reproducibility and the accuracy of the VAF that was returned. So that was really useful in retrospect to be able to have these 10 acceptance criteria, and actually we passed on all counts. So we're very lucky to have a huge base of samples that we can use retrospectively to draw upon for our validations. And we've actually put 204 unique samples through, although since then we've now sequenced well over 2,000. But this truth set included, as I've mentioned, some suboptimal samples, samples from across the region, different DNA extraction formats, and some poor quality samples. We've also been able to ensure that all of the key mutations that we're looking for were detectable. So we've included a variety of mutations across the whole spectrum of genes, which we've included in the panel. Just a quick look at coverage, and this is something we're particularly impressed with. The chip that we're using allows us to return approximately 90 million reads. So if we put 100 samples on there, 96 samples, you're looking at almost 1 million reads per patient, and then you can divide that by the number of amplicons you've got, and you can see here the one we're really interested in was exon 14, where the V617 mutation is. We're getting 30,000 reads on average, and even our poorest performing samples would have 2,000 reads, and that allowed us to confidently assert that we could reach that 1% limit of detection. So we've actually set 2,000 reads as a minimum threshold for our quality control procedures when we analyze the data. I think this is a really nice demonstration of the coverage that we got, but what I can't give you an answer for today is why, when we increase the loading mass, we actually get better coverage of individual amplicons. This is despite the fact that there's a step within the protocol where you dilute your library down to 50 picomolar, which is your optimum sequencing concentration. Even though we do that, we still see that with a higher loading mass, we get much better coverage across the amplicons. And we've settled with a 30 nanogram load for our particular assay after playing around with the parameters on the PCR. This gives a nice demonstration of the distribution of those reads across the amplicon. So it's a really lovely design. We've got a fairly even distribution. And even the poorest performing amplicon in there still gets 2% of those reads. So we've found that actually the design of this is really nice. It allows us to get very even coverage between samples and across amplicons. And I'll show you that in just a second. Something we're especially impressed with is the ability of this particular panel to sequence GC-rich regions. So the hotspots in Mipple that I've mentioned previously, that's a particularly GC-rich region, and other assays that we trialed at the time actually didn't work for Mipple at all. This assay is so good with GC-rich regions that we've actually decided to move some of our other amplicons from our pan-hemonc assay, which really struggle with a gene called CBP alpha. We're hoping that we can try and sequence this using this platform because we get some really nice coverage, even when you've got particularly GC-rich regions, which are notoriously difficult to sequence. Relative to our standard of care test, so this is the high throughput, the pan-hemonc NGS assay that we use for our acute diseases. We found that we had practically 100% sensitivity. There were some variants that weren't detected at the end of the pipeline, but that's due to them being slightly below the reporting threshold. So really, the sensitivity, it's just a case of tweaking your parameters on your pipeline. And we found that actually in the variant call files, those variants were recorded. It's just that we'd set our parameters not to return them because they were slightly below our limit of detection. And you'll remember I said we'd included some samples which had very low level variants included. So we were really testing the lower limits of the detection for this test. This is versus standard of care, so the PCR-based test that we use. We did detect a single false negative. That's because it's very likely that this sample actually has a variant which is below the limit that we're actually trying to detect. So we've now validated an additional ddPCR assay, which can support us in our confirmations, which gives a very accurate VAF. And this particular sample has a VAF around 0.4%. So the pipeline that we've designed returns everything that it finds at that position above 0.5%. And it's inevitable with any assay that there will be samples which have a VAF that hovers around your limit of detection, which on the first pass you might find at 0.4 and on the second pass you find at 0.6. What's important to point out is that this really isn't a false negative because we're only trying to detect samples above 1%. So that explains that false negative. But really, the sensitivity and the specificity was very, very good compared to our standard of care data, both from sequencing and from the targeted PCR-based test. As I'd said, we wanted to look at that V617F position in real fine detail and determine the amount of background noise we got for the wild type base, but also for the two alternative bases that you might find. And you can see there in column A, that's the normal or wild type signal. That's what we would determine as background noise. And actually, for the true signal, everything was at least 2 to 5-fold higher than the background noise. So since bringing this to service, we haven't actually had a single false positive come through, and we've got this DDR PCR test that we're able to use for confirmations of those very low level samples that return a positive result. It's been so good actually that we're probably going to retire that ddPCR based test because we just don't need it. If our NGSA returns a variant at 0.6%, the ddPCR tells us it comes through at 0.6%. So we're very happy with the background noise on this, which was our main concern, I think, moving to a PCR based NGS assay. And these results are reproducible across runs. Excuse me. So we repeated our chips 4 times, I think, and the only discrepant results were explainable either because they fell slightly below or above that 0.5% threshold that we'd asked the pipeline to return the variants for, or because the annotation scripts would annotate slightly differently for the C dot nomenclature versus the G dot nomenclature. This isn't a problem because we still get the result, but we could explain any discrepant results either because it was right at the lower limit of detection or because there was a discrepancy in the nomenclature. So there was nothing untoward with the reproducibility across runs. Comparing the VAFs that the assay returns, we've compared this to our standard of care high throughput, the pan-hemonc assay that I've mentioned previously, and we find a really tight correlation between the VAF for the standard of care sequencing versus the MPN panel. So we're very confident that the VAFs that we return are true to the true VAF of the sample. We've also included in this, remember, some validated control samples, and we found that there was a very precise correlation between the VAF of those control samples and the VAF that the panel returned. So I'll just move on quickly now to talk about, in the last few minutes, the design of the service for the patients. As I said, we're looking at approximately 200 samples a week, and then a little bit of the audit data that we've got. So we have a 6-day rolling schedule in the lab. We start on Mondays, get the samples out, and we do our PCR. We spend Tuesdays cleaning up the libraries and quantifying them and putting them onto the chip, and then sequencing happens on day 3. That's fairly quick, relatively. I think it's 3 or 4 hours for the sequencing. Because we've got 100 samples on there, when we put them on our postscript pipeline, that can take 11 or 12 hours, but the sequencing is all complete within a couple of hours. So then we start the postscripts and we run that overnight. That gives us the ability that if a sample does fail, we can then get it onto the next run. So we have 24 hours in between run 1 finishing and run 2 starting on the Thursday to get any failed samples on. And that means that we can get a very tight turnaround time. So you can see the longest predicted turnaround time for a sample which failed, because it can go straight onto the next run, would be 14 days. And I'll show you some audit data in a moment, just how that looks in service. These were the QC thresholds that we determined. So we said that every chip should have a minimum of 50 million reads returned. As I've said, that we've been getting up to 90 million reads. Each sample should have a minimum of 100,000 reads in order to pass QC. And this is a typical run from a couple of weeks ago. The reason those samples are also nicely distributed is because we've quantified them using the qPCR kit that comes with the assay. And that allows us to pool our samples in a normalized fashion. And you can see here we've had 2 failed samples, but everything else has achieved between 2 and 5 times as many reads as we would need that particular sample to get to pass QC. So we get a really nice distribution of reads and well above what our minimum is to pass QC thresholds. So we're at 6 months now. We've done 60 separate chips. We have had 2 chips fail, but both of those were user error. We've introduced a new step in the process to check that all of the caps have been removed from the reagents. Obviously, the robots don't like it if there's a cap still on the tube, but the only runs that have failed have been due to user error on our part. And as I mentioned, we're hitting consistently 85 million reads, averaging at 87, and the highest I've seen so far a few weeks ago was 92 million reads. So I'm not sure we can go much higher than that, but we definitely have spare reads available. We're clearly surpassing the requirements that we have for QC. We've also got some really good bioinformaticians on board, and they've been able to write a program that allows us to produce PDF reports for the clinicians. So if a sample has one of the hotspot variants that we've discussed, and it's above the limit of detection of the test, it will write the report for us. So that only needs one scientist to then come in and check the report, check the patient details, and make sure that everything's correct. And we can actually get those reports out on average within 10 days. So that's from the sample being received in the lab. And if you contrast this to the previous standard of care, where we had disparate turnaround times, and some places had an average of 21 days, this is a significant improvement for us. The failure rate I mentioned was very, very low. We had allowed 24 hours in between runs so that we could get the failed samples back onto the next run. But as it happens, we just don't have enough samples to justify waiting an extra day that fail. So it's likely that we can actually squeeze this down and take another day off that turnaround time, because we just aren't seeing that many fails to warrant holding things up for an extra day. So that's all looking really good, and the feedback from the clinicians have been great. I'll just finish off in the last minute or two with a couple of interesting case studies. Previously, we would have only looked for a JAK2 mutation in the first instance, and this patient that you can see here, we've detected on the panel another mutation in the MPL gene, the thrombopoietin receptor. So this is a bit of an unknown entity. We don't know how many of these patients are out there that have two mutations. If we'd found a JAK2, we would have given a diagnosis and stopped testing at that point. So this may be a new class of a new group of patients that have different clinical outcomes. So it's really interesting to us now that we're finding these double mutations. We've also picked up a whole host of atypical JAK2 driver mutations, including some which produce a V617F mutation, but which have a slightly different CDOT nomenclature. And these wouldn't have worked previously with our old PCR-based test, where the primers wouldn't have bound if it wasn't the standard mutation. So that's a lovely outcome from the panel validation. And I think this is probably the story that I'm most keen to tell. Previously, with the 15% limit of detection for Sanger sequencing, those patients would have had to have had a bone marrow biopsy to get their diagnosis. But with the improved limit of detection with the NGS assay, we're now able to detect this down to 5%. And this is from the last 6 months, we've got 10 or 11 patients who we've found a diagnostic variant, and we've been able to give the diagnosis without the need for them to have a bone marrow biopsy. Once you start thinking about the cost of theatre assistance, anaesthetic, even though it's a fairly minor operation, and obviously from the patient's perspective, having a bone marrow biopsy is something they would avoid at all costs if possible. Some of them will have to have their disease staged by looking at the marrow to see if there's fibrosis, but we do know that there's a subset of patients that will be able to avoid having a bone marrow sampling done, which is a great success for us. That very rare entity, chronic neutrophilic leukemia that I mentioned, which is characterized by a CSF3R mutation, we've picked up one of those. This actually came in from a very old female patient who presented to her GP. So she would have had a fairly long diagnostic process to get this diagnosis delivered, but we've picked this up in the first instance on a peripheral blood sample from a GP. So that's a really interesting story to tell as well. So I'll just quickly summarize this. NGS mini panel for MPN has clearly improved and standardized the limit of detection. We can pick all of our variants up, and all of the patients get exactly the same limit of detection now. So it doesn't depend on which type of mutation you've got for how long it takes to diagnose you. We've improved the turnaround time from 21 days to 10 days on average. As I mentioned, and I'm really proud of, that fewer patients are having to have bone marrow samples. We've got new categories where we're finding rare drivers. And I think this is really nice because we've managed to retire some of the old tests and we've consolidated our testing into a single center. We're actually delivering this NGS panel for approximately the same cost as we were delivering our previous standard of care. So with that, I'd just like to wrap up and say thank you very much to the team in HMDS, particularly the bioinformaticians who've done a fantastic job working up this panel for us. So thank you very much, and I'll be sticking around to answer a few questions if anyone has any. Thank you.